Hello everyone, I welcome you all to today's session. I am Shail and let's compare DNA polymerase 1 with 3 found in prokaryotes during replication. Both have polymerizing activity in 5' to 3' direction. Both have proofreading activity, that is 3' to 5' exonuclease activity. But DNA polymerase 3 is highly processive, that is, once it attaches with the template strand, it synthesizes many nucleotides before detaching itself from it, which is lacking in case of DNA polymerase 1, that is, its processivity is very low, because of which 3 acts as the main polymerizing enzyme, whereas 1 is used to remove primers, because 1 has one extra special activity, that is, it has 5' to 3' exonuclease activity. And the fragment because of which DNA polymerase 1 acts this way is called as Klenow fragment, and because of that it can be also called as Klenow enzyme. Thank you for watching.